Welcome, everybody. Thank you for coming. My name is Chris Leonard, and it is absolutely my pleasure and honor to be able to welcome you to the 12th annual Gabor and Harriette Kaley Endowed Lectureship. Today's speaker is Dr. Constantino Iadecola from Cornell, and he will be properly introduced in just a few moments. What I wanted to say is the lectureship was established by the generosity of Dr. Harriette Kaley and her son David Kaley to help us remember the great contributions Gabor Kaley made to this institution and to physiology in general. And he was the chairman of the physiology department for almost 40 years, which was an amazingly fertile period of time for that department. There were a number of program project grants. They really established bona fides in international recognition for their work in cardiovascular and microcirculation. And it's really a delight that we have Dr. Harriette Kaley here with us as well, and she'll say a few words in a moment. I was one of those people who was lucky enough to know Gabor, and he was a spectacular scientist, inspiring, and he was a great leader. He was really a terrific chairman of the department, and his contributions are worth recognizing as often as possible because it helps us know the goals that we should set for our own professional development and careers. So it's inspiring to be able to recognize him on an annual basis. It's also this year auspicious, even though this is the 12th time we've done this, this is the first time we've done it in the evolved form of the physiology department. Physiology has been combined with two other departments, and it's now the Department of Cell and Molecular Physiology. And I hope that going forward, that we will live up to the success of the prior era of the or prior Kaley era in the Department of Physiology. So just before I introduce Dr. Harriette Kaley, I just want to thank all the people that have gone into making this successful. I want to thank our AV crew, who I see some of them are still here. That's very good. That includes Juan Castro, Michael Frankfurter, and Corinne Zasterchek. And I also want to thank the selection committee, the board that selected today's speaker and previous speakers. That included Dr. Mike Wolin as the head of that committee, Dr. Rhoda, and several other faculty that have been intimately involved in fleshing out various selective candidates. So how's this going to go? I'm going to introduce Dr. Kaley, then Belinda will introduce Dr. Iadecola, and Dr. Iadecola will give his talk if there's any time left. And after that, we'll have some questions and answers, which will be moderated by Dr. Berry. And then will be some closing remarks by the chairman of our department, Dr. Joe Entlinger. And after that, we will retire for lunch. So, oh no, we have an award ceremony. How silly of me. We have an award ceremony. Yes, okay. So in any case, it's a pleasure to introduce Dr. Harriette Kaley, who will say a few words in remembrance of Gabor. Dr. Harriette Kaley is a psychotherapist and has been sorry. Well, in that case, everything else I'm going to say is meaningless. But no, she's a psychoanalyst and has been an inspiration to Gabor all of his modern professional career, and has been an inspiration to all of us who know her as well. Thank you.
I want to say I'm looking at the captions, and I'm tickled to see that whoever is doing this or whatever is doing this is having the same trouble we always had with the spelling of our name. You notice it varies every time it comes up. My favorite was there were all kinds of variations on it. My favorite was Dr. Gailey Kaley. So all I really want to say is that I'm so gratified that this event is happening, that it's the 12th year, a dozen years. Gabe would have been so happy and so proud. And I'm just touched to see you all here. Could I, just for my own personal satisfaction, could I see how many of you actually met him, knew him? Yay. There are fewer of us. We have to keep on remembering. That's all I really wanted to say. It's just very gratifying. Gabe wanted this lectureship to be established. He wanted to keep on teaching. And I'm so touched and honored. And I know David would be also, our son. He happens to be in Korea right now working. So I'm here to speak for him also. Thank you all for being here. And I'm looking forward to what will be another interesting lecture, which even a psychologist like me can sometimes understand snippets of. So that's good. Okay, thank you.
Dr. Kaley, thank you for joining us today and for sharing the wonderful stories about your remarkable husband. Good morning and welcome to the 12th Annual Drs. Gabor and Harriette Kaley Endowed Lectureship, hosted by the Department of Cell and Molecular Physiology. Thank you to those joining us in person and to those participating remotely. We are very delighted to see such a strong turnout. My name is Belinda Nyantaki, and it is a great honor for me to introduce Dr. Costantino Iedicola, the recipient of the 12th edition of the Drs. Gabor and Harriette Kaley Endowed Lectureship. Dr. Iedicola is the director and chair of the Fale Family Brain and Mind Research Institute at Weill Cornell Medicine. He also holds the Ann Parrish Titzel Professorship of Neurology. Dr. Iedicola received his medical degree from the Università di Roma and completed advanced training in neurology and cerebrovascular research in Italy and the United States. Dr. Iedicola is internationally recognized as a pioneer in cerebrovascular biology and neurovascular research. He has made seminal contributions that have transformed our understanding of key mechanisms underlying neurovascular coupling and vascular cognitive impairment. His work has elucidated how vascular dysfunction, inflammation, immune-brain interactions, and metabolic factors contribute to both acute brain injury and neurodegeneration. Over the course of his career, Dr. Iedicola has authored hundreds of peer-reviewed publications in leading journals such as Nature, Neuron, Science, and the Journal of Neuroscience. His research has been continuously supported by the National Institute of Health, the American Heart Association, and major Alzheimer's disease foundations. Dr. Iedicola's leadership extends well beyond his laboratory. He has played and continues to play a central role in shaping the field through service on numerous advisory committees, including those of the NIH and the American Heart Association. He has also chaired or contributed to influential scientific statements and guidelines on stroke, hypertension, and brain health. His many prestigious honors include multiple NIH Javits Awards, the American Heart Association Distinguished Scientist Award, and the Lifetime Achievement Award from the International Society of Cerebral Blood Flow and Metabolism. Today, Dr. Iedicola will share insights from his groundbreaking research on neurovascular dysfunction. Please join me in welcoming Dr. Costantino Yedicola.
It's really an honor to be here today and participate in this annual event, which is sponsored by the memory and by Dr. Kaley, who's present here. And when I found out that she was a psychoanalyst, I stopped talking because, you know, I didn't want to reveal all my, what went on in my, and talking about my, you know, mispelling of the, I mean, mispronouncing of the name, the best one that I remember is Ayatollah. You know, Yadekola, you know, Ayatollah. So that was the, so we're all on the same, you know, although your name is much easier than mine. Anyway. So thank you again. It's nice to have you here. And I hope that I will be able to tell you a little bit about what we're doing in the general context of what's going on with the brain, with our brains when we get old, all right? And first I'm going to scare you. Okay, dementia is the only disease, major disease, in which the mortality has been increasing over the years. So here you can see from the changes in selected causes of death, you know, according to US statistics from in the past 20 years or so. And as you can see, there is the only disease where there's been such a radical increase has been Alzheimer's disease. You can say dementia in general, dementing cognitive impairment and so on, okay? And because of the aging of the US population, and actually for that matter, the world population, what we're gonna have is that right now we have about 8 million people in the US with Alzheimer's disease. So in the next few, you know, decades, this number is gonna double. And the cost of this doubling is gonna be exceptional because people live longer, they require all kinds of assistance. There is stress on the families and the caretakers and so on. So it's becoming one of the major problems affecting human health in general, you know, because it's such a prolonged condition for such an extended period of time. So that's why today I'm going to tell you a little bit about how the concept of dementia to the ages and how the whole thing evolved. The way we see dementia has changed over the past 100 years or so. And Dr. Kaley would be happy your husband would be happy to be here, I guess. Maybe but also in spirit, I guess, because the vessels have really found their way back into the brain in a major way that would never been appreciated at the time, you know, in the 70s, 80s, and 90s, and so on. And they are here to stay across the entire spectrum of brain diseases affecting cognition. We're going to talk a little bit about the role of blood vessels in keeping the brain going and what happens when things don't go well and what are the major culprits of these things not going well. We're going to talk about amyloid beta, which is one of the major factors in Alzheimer's disease, hypertension, and ApoE4 genotype, which is the major risk factor for cognitive impairment, period. And finally, we're going to see how the whole thing can come together in this recently established treatment for Alzheimer's disease, which has made a major impact worldwide, which is the use of A-beta immunotherapy. And we will see how what I told you about the blood vessels of the brain, how relevant it is for this new development that we have been experiencing. So let's look at the concept of dementia. At the time of Alzheimer, okay, so we're talking about the turn of the 19th to 20th century, all right? The major cause of dementia was thought to be hardening of the arteries. If we would call that vascular dementia today. They thought was due to atherosclerosis of the blood vessels of the brain getting hard, and then the brain didn't get enough blood. And there was a starvation process of the brain leading to cognitive impairment, okay? At the time, as we have today, the dementia was divided in two different categories. One was pre-senile dementia for people who were younger than 65 years old, and senile dementia for people who were older than 65. So vascular dementia was felt to be the major cause of senile dementia, all right? But then he described this youngish woman, middle-aged woman, okay, who was profoundly demented when she was like 45, 50 years old, all right? And he described the pathology of this condition, so-called Alzheimer, which then became known as the Alzheimer pathology, which is amyloid plaques and neurofibrillary tangles. And Alzheimer's disease, eventually was established as the major cause of pre-senile dementia. So dementia of people younger than 65. So it was not really all that important, okay? Because few people experience dementia when they are so young. Most people develop dementia when they are old. So for many decades, Alzheimer's disease remained an interesting thing, but was not really of all the major clinical interest because it was really in a restricted population, a younger population. Now, the first conceptual shift in the concept of dementia caused by vascular lesions was provided by Vladimir Ashinsky, which is a fantastic Canadian neurologist who's made a major impact on world neurology. So Ashinsky, based upon pathology work done by pathologists in England, he said that Alzheimer was wrong. So hardening of the arteries is not the cause of vascular cognitive impairment. But the reason why people get demented when they have vascular problems is because they have multiple strokes. So one stroke after the other eventually would kind of rotten the brain. So they would become them. He called that multi-infarct dementia. And you can see here with the CAT scan, I mean, this is an MRI, but you can see the multiple holes in the brain in a case of vascular cognitive impairment. However, when the CAT scan was introduced in the 70s and so on, it became obvious that very, very few people have multi-infarct dementia. Okay, so multi-infarct dementia could not be really the major cause of cognitive impairment. But the conceptual advance was that with Ashinsky's proposal, dementia became a preventable disease because at the time they already knew how to prevent strokes. So his idea was that by preventing strokes, you can prevent dementia. And Ashinsky was fortunately still alive. He is pushing that concept even today, which is not all that widely accepted in a more kind of neurodegenerative disease circles, all right? So the advance was more conceptual than actually, nosological. Now, almost at the same time, in the late 60s, early 70s, those guys in England, the pathologists, kept studying these old brains, all right? And what they found was that, in this paper published in Nature, all right, they found that in people with senile dementia, so old people, 70, 80-year-old with dementia, they had a lot of Alzheimer-like pathology. So as you can see here, this is the score of them, how demented they were. The more plaques they had, the more demented they were. So this data was the beginning of Alzheimer taking over the entire field of dementia. So from a pre-senile, nickel and dime kind of neurological condition became the prevailing cause, or at least was appreciated as the prevailing cause of dementia, all right? And then, the things move forward, the amyloid plaques were identified as being made by a peptide, amyloid beta, that comes from a protein called the amyloid precursor protein, okay? And so the molecular pathology, essentially, of the plaques was developed. The same thing happened with the neurofibrillary tangles, with the discovery of tau synthesizing enzymes and phosphorylating enzymes and so on. And eventually, the concept of the so-called amyloid cascade hypothesis was put forward as the major cause of Alzheimer's disease. okay? Hence, the major cause of dementia in the elderly, all right? Because of that what I told you about this shift between presenile and senile, all right? So Alzheimer, Achinsky called this process, Alzheimerization of dementia, okay? Now, if you give a talk to a lay group and you say, how many of you know what dementia is? You're going to have a couple of smart little ladies, raising their hand. If you say, how many of you know what Alzheimer is? Everybody raises their hand, all right? Because the concept of dementia has been captured by the Alzheimer phenotype, all right? But things turn out to be different than that. Over the past 3 decades, it's become obvious that Alzheimer's disease is not caused only by plaques and tangles, or what Alzheimer thought to be the cause of the problem, but there are different pathologies causing the dementia, and the majority of which is vascular. So in community-based dementia cohorts, you will find that the major cause of cognitive impairment is not Alzheimer pathology, which is plaques or tangles, but is a mixture of a variety of pathology of which the vessels play a major role. And this, you may argue that this is kind of pathology data, so kind of end of the line, burned out disease, all right? But recent proteomic studies of the CSF, cerebrospinal fluid, when you do a spinal tap and you get the stuff out, they've shown that, in Alzheimer's disease, in what's called clinically Alzheimer's disease, you have 4 or 5, 6 different kind of groups of diseases: vascular, RNA dysregulation, hyperplasticity, immune factors, even the choroidal plexus that has been implicated. So as you can see, you're talking about really a syndrome in which you have a wide variety of pathologies that may be more prevalent in one group versus another group. But certainly, the vessels are a major component. And ironically, one of the latest proteomic studies done in a community cohort of patients with dementia, with so-called Alzheimer's disease, all right? They found that hardening of the arteries of Alzheimer, which is, intracranial atherosclerosis, was an independent pathogenic factor in Alzheimer's disease. So back to the future, all right, back to what Alzheimer was believing, the latest data shows the importance of the vessels of the brain in maintaining brain health, cognitive health of the brain. So the idea that we were taught, in medical school, now I'm not going to tell you when, but you can guess that you had, vascular diseases of the brain causing, cognitive impairment by killing the brain by strokes or whatever. And then you had neurodegeneration, which is completely different, all right? It's no longer tenable because there is an interaction between these factors so that, in a community-based cohort, the major problem is not Alzheimer pathology, is not vascular pathology, but is a series of pathology together, of which Alzheimer and vascular are an important component, all right? So things have gotten more complicated from a single thing that they maybe was taught before to multiplicity of factors, which has profound implications, as we will see, for therapy, all right? Because you cannot treat the same patients with the same drugs or whatever, because each one is going to be a little bit different, having a prevailing pathology, all right? A little bit more about that. Now, as Dr. Kaley would have told you, all right, nobody should be surprised that if the vessels of the brain don't work, the brain doesn't work either, all right? The brain is supplied by 4 separate blood vessels running through the neck, and converging at the base of the brain in what's called the circle of Willis, which is here at the base of the brain, from where all the vessels vascularizing the brain originate. These vessels wrap around the brain from the outside in, and they dive in a vascular network, which is estimated to be 400 miles, in a volume of a bottle of wine, 750 milliliters, all right? So you can imagine, you know, how much stuff is, is pressed in there, all right? Very, very tight, all right? Now, these vessels do a lot for the brain, more than we thought, more than at the time that Dr. Kaley was active, you know, he would have thought. For example, we know that the vessels of the brain are important for feeding, providing oxygen and glucose, which is the fuel of the brain, commensurate to the level of activity that the brain is at, all right? So if you're, you know, resting quietly in the chair, you're not going to need as much oxygen and glucose in your brain if you're, you know, solving a complex equation, all right? So there is the more blood is needed because neurons are active and they need to be fed, all right, as they're consuming ATP to create their energy. And so the vessels do that. So neural activity tells the blood vessels, I need more blood, and the vessels comply by dilating, reducing vascular resistance, and flooding the active territory with more blood, all right? Now, the endothelium, which is one of the favorite subjects of Dr. Kaley's work, also is critical for the brain. As Dr. Kaley showed, you know, the endothelium makes vasoactive factors like prostanoids, nitric oxide, all right, constricting factors like endothelin, all right? So it can regulate vascular tone independently of everything else. But the brain endothelium also is responsible for the blood-brain barrier. So the brain doesn't let anything from the blood in that doesn't want, all right? And the process of transfer from the blood to the brain and from the brain to the blood is very controlled by transporters, by molecular transport systems that determines what goes in and what comes out of the brain. And that's called the blood-brain barrier. And the essence of the blood-brain barrier is the cerebral endothelium. One thing which has come out over the past 10, 15 years is that the brain vessels are also critical for removing the garbage that the brain generates as a result of its activity. So the brain is one of the most metabolically active organs, and it generates a lot of junk, proteins that the brain doesn't want to keep around. So which one are these proteins? A beta is made by synaptic activity, and you got to get rid of it, all right? Tau is made by synaptic activity. So when the neurons talk to each other, they make a beta and tau, all right? I thought that was very, very surprising. And why don't we get Alzheimer's disease when we're, you know, 25 years old? Well, because the red blood vessels get rid of them through a process of clearance, okay? Now, as we were discussing earlier, there is a controversy. How does the brain get rid of all this junk that is being made, all right? But we all agree that the brain does get rid of it, okay? Because these proteins need to be removed because they are called disordered. So disordered protein means that they do not have a very specific conformational shape, so they can kind of easily aggregate, you know, and get stuck into places we don't want it to be, like A beta in the extracellular space or tau into the neurons themselves, all right? So you have to get rid of it to allow that the concentrations get high enough to cause their aggregation. And the vessels do that, okay? And just to throw some names in there, so there is the so-called lymphatic hypothesis of brain clearance, where the stuff moves forward to the perivascular space into the venous side. There is the retrograde perivascular hypothesis, whereby the stuff moves backward and gets into the subarachnoid space and eventually is going to get clear. And there is also something called mixing, ISF, CSF mixing, where the stuff just mixes together. And so the concentration gets diluted and removed from this way from the brain. Finally, these perivascular spaces and the meninges and so on, they are a very critically important immune organ, all right? The brain was said to be immune privileged, all right? And that's, now we don't think that the brain is immune privileged anymore. T cells, lymphocytes, monocytes, neutrophils, they easily get in and out of the brain at their will in a very orchestrated manner through specific pathways, which are all perivascular. Okay, so the vessels are also the conduit, the space around the vessel, where these cells traffic in and out. When things go bad, they play a major role in disease. And we're going to see a little bit an example of that later on in the talk, all right? So considering all the stuff that the vessels do for the brain, okay, it's not surprising that when the factors that affect vascular function will lead to brain dysfunction. And brain dysfunction underlies cognitive impairment, because the function of the brain that requires all the parts of the brain to work in concert is cognition, all right? If the brain doesn't work right, if the right end doesn't talk to the left end and so on, all you're going to get is cognitive impairment. Okay, so you need the perfect working of all those things. You need the clearance. You need the delivery of ATP and glucose and oxygen. You need the endothelium to do whatever it does, the blood-brain barrier and so on. You need all those things to work in harmony to have a healthy brain. All right. So let's talk about amyloid beta, which is one of the major factors affecting, responsible for Alzheimer's disease. So the traditional view, which I do not question at all, is that amyloid beta, or perhaps tau, they directly disrupt the communication between neurons, synaptic activity, all right? They're very well known to do that. However, the data that recently accumulated concerning the role of vascular factors in Alzheimer raised the possibility that amyloid beta, in addition to acting directly on neurons, may also mess up the blood vessels. In which case, you would have kind of a feedback mechanism whereby not only there is a direct neuronal dysfunction, but also there is vascular dysfunction contributing to the dysfunction of the neurons, all right? Now, this concept was was proposed years and years ago, all right? But could not be proven until mouse model of Alzheimer's disease were developed. Because the studies on Alzheimer, at least particularly in the old days, were mainly done on pathologist specimens. And so the neurodegeneration people would say, well, the vessels are not right because, the neurons have died, so the vessels are also dying, all right? So there was no demonstration that actually vascular dysfunction was occurring early in that brain to contribute to the pathology. So we had the opportunity early in the game to look at mouse models in which amyloid was accumulating. And we studied these mice before they got sick to try to figure out whether vascular dysfunction was an early manifestation of the A-beta accumulation process. So we took these mice and we measured, the blood flow increases induced by neural activity. We topically applied on the brain acetylcholine, which is a node-generating compound when put on the brain vessels as well as in isolated vessels. So it activates cholinergic receptors leading to the release of nitric oxide, endothelial nitric oxide, to study these aspects of the regulation of cerebral circulation, the endothelial function as well as the neurovascular coupling function. And what we found was that these mice, when they were otherwise healthy, so before they got sick, they already had profound attenuation of these fundamental responses of the cerebral circulation. When this paper was published in the late 90s, people was completely disregarded until human study showed that the first thing that goes wrong in an Alzheimer brain is vascular dysfunction. And then, since then, there has been a major, what I call a bandwagon, of interest, oh, everything now is neurovascular, all right? Neurovascular this, neurovascular that. Pericytes do this, Alzheimer is caused by pericytis. So there has been this big kind of, confusing and often, not accurate role of blood vessels because people tend to exaggerate, all right? So Alzheimer is not a vascular disease. It's a component of the process. In some patients is vascular. That's the proper way to put it, all right? It's not stroke, all right? But nevertheless, vessels, this data shows that vessels play a role early in the disease process, okay? Now, how that happens took a little bit of playing, to figure it out. So A beta seems to kind of bind to a receptor, leading to activation of inflammation and oxidative stress through an enzyme called NADPH oxidase, and not particularly NOX2, that is a major generator of free radicals, and leading to eventually a calcium overload of vascular cells, making it unable to work right. Now, as many of you in the audience know, for many, many years, there has been a controversy about where the radicals come from in the vasculature. Some people say it's the endothelium, some people say the smooth muscle cells, but those cells are not really professional free radical producers. And what we found was that the major source of radicals in many pathological conditions I'm going to tell you today is not the vascular cells in themselves, but they are immune cells closely associated with blood vessels. And these cells are called perivascular macrophages. So what are these perivascular macrophages? So they are one of the group of the brain resident macrophages that originate from the yolk sac very early in development. They reach the brain as erythromyeloid precursors, and they give rise to the four major classes of immune cells, innate immune cells in the brain. And you can see them here. The microglia, which is very popular these days, the meningeal macrophages, the perivascular macrophages that you see here, and the choroplexus macrophages. So these cells, they look exactly like macrophages, except they are plastered on the external surface of the blood vessels. So, and they can make huge amounts of radicals because that's all they do. They phagocyte stuff and they make radicals to destroy, germs or whatever they are. And now you can imagine if you have this profound source of radical next to a vessel that doesn't like radicals at all, you're going to have a problem. And we found that in A beta pathology, this was really the problem. Now, how do we get there? Okay, how do we get to the conclusion that this amyloid beta response was mediated by these macrophages? Well, one way we did was to kill all the macrophages in the brain, specifically the one around the blood vessels, all right, using something called clodronate, encapsulated into liposomes. So when you do that, and you inject it into the ventricles of the brain, the phagocytic cells within the brain spaces, pick it up, all right? The lipids come off, and clodronate becomes a toxin for the macrophage. It's like a Trojan horse type of thing, all right? They bring it in, and then they die from it. And if you do that, you can see here the macrophages populating the perivascular spaces, after clodronate, you don't see anything. And you don't see any effects on the peripheral cells because we injected the drug into the brain, okay? And there is nothing going on with the microglia because microglia in a resting state are not phagocytic. So they don't pick up the stuff, all right? And then if you do that, and you look at the effects of amyloid, they are completely reversed. You can see here the response to neurovascular coupling is reversed. The response to the endothelium, the radical production is no longer there, while A beta is still there, all right? So A beta is present, but the target of A beta, which is this perivascular macrophage, is not. So there is no production of radicals, and there is a normal neurovascular function. Now, this was a kind of the shotgun approach to look at this, because you just get rid of the cell. But what we wanted to know, what about we want to get rid of the receptor for A beta on these cells, which is CD36, all right? And we want to get rid of NOX2 from these cells, which is the source of the radicals. How do we do that? One way to do that is to do a bone marrow transplant. So if you transplant a bone marrow into a wild type animal, all right? Let's say in this case is a bone marrow expressing GFP. After about 3 or 4 weeks, the perivascular macrophages become GFP positive, which means with the radiation of the brain, you have removed all the macrophages, which are very sensitive to radiation, and these are replaced by bone marrow derived cells that take the place of those we have gotten rid of, all right? So full disclosure, the cells are not the same that the mouse came with, right? They are coming from the bone marrow. But that's the best thing we could do at the time. I'll show you later a better way to do it, but just for the flow of the science, all right? So if you transplant the bone marrow that lacks the receptor for A beta, you can have a transgenic animal who has no receptor for A beta in the perivascular macrophages. And if you transplant a NOX2 more deficient bone marrow in the same mice, you're going to have perivascular macrophages without it, and that's exactly what happened. So if we transplanted either the NOX2 negative marrow or the receptor negative marrow, we could completely rescue the increases in blood flow produced by functional hyperemia without affecting the levels of amyloid beta. All right. So amyloid beta is still there. The macrophages are still there, but they do not have the critical NOX2 and CD36 to mediate this neurovascular dysfunction. Now, those mice that I told you so far are young mice, all right? They don't have any cognitive impairment. And you may wonder, what happened when the brain gets pathologically affected by the pathology, by the plaques and everything? So we aged those mice, and we found that if we did the bone marrow transplant business, we could rescue this functional hyperemia and also cognitive function in a mouse by removing these receptors from the macrophages. Now, how do you assess cognitive impairment in a mouse? Make some people laugh, all right? But people, the community wants us to study cognition in a mouse, all right? So the test that I love, that I prefer, is called nesting, which is the ability of the mouse to build a nest. Male mouse or female mouse love to build nests. So if you give them a bunch of white paper in a cage, okay, they neatly disperse like that, all right? They neatly assemble it into a nest. So which is an activity of daily living for the mouse, okay? So if they get Alzheimer, all right, they lose the ability to build a nest, okay? And that's used as an index of cognitive impairment, all right? That's always tricky when you present to neurologists, because they all start laughing, what does it mean? But I say it's an activity of daily living, all right? Anyway, but what happened to the brain of these mice is very interesting. So when we removed the A beta receptor, we found that the amount of A beta associated with the blood vessels is much reduced, shown by several quantitative measures, all right? But the plaques, which is the amount of plaques within the brain parenchyma, is still there. So by removing this receptor, we were able to reduce the amount of A beta associated with the blood vessel without affecting the amount of A beta deposited in the parenchyma. But the mouse were cognitively normal, all right? So highlighting the fact that the vascular associated A beta is very bad for cognition, worse than what A beta in the parenchyma does, as shown here, all right? As shown here. Now, the fact that by removing this receptor, reduce the A beta around the blood vessels, raise the possibility that when you have this receptor impairs the ability of the vessels to get rid of A beta. Okay, that's why it gets stuck in there when you have in the presence of the receptor. So we measure the clearance of A beta in mice lacking the receptor and see if that would be better or worse. So how do we do that? Well, one of the clearance pathway is through the endothelium. It's called transvascular pathway. So A beta from the brain gets directly into the blood via an endothelium transport system. Is to pick that up, you have to measure the A beta coming out of the brain from the superior sagittal sinus, which is where all the venous outflow of the cortex comes from. All right? Then we measure how much A beta was left into the brain and how much A beta reached the blood. All right? So if the mouse did not have the receptor, they were able to retain less A beta in the brain, so they got rid of it very well, better. They had more A beta coming out of the superior sagittal sinus, and they had more A beta in the circulation. So by removing this receptor, the ability of the brain to clear A beta was improved. So to put it all together then, what happens in these models is that A beta acts on the perivascular macrophage to induce free radical and inflammation, which paralyzes the blood vessel and impairs the ability of the vessel to get rid of amyloid. And as a result of that, you have accumulation of amyloid around the blood vessel. It's called cerebral amyloid angiopathy. Okay? So the macrophages are really critical for this accumulation of A beta around the blood vessels. Now, hypertension is another big cause of cognitive impairment. Not only is a major cause of stroke, one of the major risk factors for stroke, but it's also a major risk factor for Alzheimer's disease, doubling the incidence of Alzheimer's. Midlife hypertension doubles the risk of Alzheimer's disease later in life, all right? So how does that happen? That has been always a question. How does hypertension lead to this? So we use the model of hypertension called the Doka-Salt hypertension model, where you kind of restrict the ability of the mouse to get rid of sodium. Okay, and as a result of that, you have an expansion of the volume of the blood volume that increases blood pressure, and also you have sympathetic activation that contributes to lowering blood pressure. So you can see here that this is the blood pressure. This is Monica Santisteban did this work before she moved to Vanderbilt. Systolic blood pressure, goes up in this as you have this model in place. But also you can see that functional hyperemia is attenuated, the ability of the vessel to increase blood flow during neural activity, and also the ability of the vessel to dilate in response to acetylcholine, which is nitric oxide dependent phenomenon, is also attenuated. So again, we have a profound neurovascular dysfunction with hypertension. And the mice became demented, all right? Again, as I told you before, both this Barnes-Maze test or this novel object, which is another way to test it, but also my favorite, the nesting that was really profoundly affected. So hypertension in this model leads to cognitive impairment like it does in humans, all right? The question is how and why? Well, we had previously found that if you feed salt to a mouse, all right, the mouse get demented, okay? If you feel, which corresponds very well to epidemiological study that, brain health depends upon the salt intake. In Japan, for example, in the 60s, there was huge increase in dementia, which was attributed to the fact that they used to eat, salt fish and all that kind of stuff, all right? Now, in these previous studies, we found that the salt led to an action on the gut that led to the production of certain cells, lymphoid cells, lymphocytes, that make a cytokine called IL-17. So we wondered if the same thing was happening in this model. So we looked at this in this Doka model, and we indeed found that in the lamina propria of the intestine, there were IL-17 producing cells when the mouse were made hypertensive. Okay, and these cells made IL-17 that you can measure it in the circulation. So they were spilling over this stuff, from the gut to the circulation. And then to find out whether this IL-17, circulating IL-17 was important, we used IL-17 knockout mice. Mice that did not have IL-17. And when we fed them the DOCA salt model, we found that they develop high blood pressure, all right, as you can see here, with the IL-17 knockout mice, but did not show any evidence of vascular dysfunction or cognitive dysfunction, despite the elevation in blood pressure. So IL-17 was really the critical factor in inducing dementia in these mice. Now, one possibility is that IL-17 acts on receptors on blood vessels in the brain, okay, which are present, known to be present, and then triggers endothelial dysfunction and everything else, all right? So we use the gene viral transfection approach to downregulate IL-17 receptors selectively from the cerebral endothelium using this BR1 virus shown over here. And we validated the method and all of that. And what we found that when we deleted the IL-17 receptors from the endothelium, we found that the endothelial vasodilation was completely restored, all right? But functional hyperemia was not. So the ability of the vessels to increase blood flow in response to neural activity was still lost, but the endothelials was working, suggesting that the IL-17 receptor in the endothelium was responsible only for an aspect of the pathological process, was not responsible for the entirety of it. So the next question is, where is IL-17 acting beyond the endothelium to cause this dysfunction? And so we looked at the possibility that the IL-17 on perivascular macrophages could be also responsible for this effect. And to do that, we developed a new mouse model that allowed us to delete selectively IL-17 receptor from perivascular macrophages using this MRC1-CRE approach. And if we did so, we rescued completely functional hyperemia while endothelial vasodilation was still depressed, all right? Why? Because circulating level of IL-17 was still increased, all right? We were only dealing with the brain side of the IL-17. So then the question is, where does the IL-17 within the brain come from, all right? There was no evidence of blood-brain barrier opening so that the one from the blood could have gotten to the brain. We couldn't find any message for IL-17 in the brain. And after struggling for months and months and months, we found that the IL-17 was coming from the meninges, of all places. We found that there was a population of IL-17 producing cells located in the meninges, which turned out to be gamma delta T cells. They were making this IL-17 into the meningeal space, okay? And we showed that using this ELISpot approach, these cells, were actually making it. It was not just positive for IL-17, but we're actively generating this IL-17. But we have a problem now because the meningeal space is separated from the subarachnoid space by the arachnoid membrane. And the arachnoid membrane has got tight junctions. What that means is that stuff that is up here, okay, in the dura, is not going to get into the subarachnoid space because of this tight junction rich arachnoid. So we had to hypothesize that hypertension was disrupting the arachnoid barrier in the meninges. And indeed, using markers of the arachnoid barrier, this Claudin 11, we found that indeed hypertension was disrupting the arachnoid barrier, allowing IL-17 to get from the dura into the subarachnoid space. And in fact, we measured IL-17 in the CSF, and we found that there was an increase in IL-17 in the CSF. So it actually percolated through the barrier to reach the perivascular space. So then the data suggests that in this model, cognitive impairment, which, by the way, salt-sensitive hypertension is one of the most common forms of hypertension in humans, all right? So it's really highly relevant to the human situation. So in this salt-sensitive hypertension, we have this production of these IL-17 cells in the small intestine that leads to an increase in circulating IL-17 that acts on IL-17 on the receptors in the endothelial cells, leading to endothelial dysfunction. At the same time, we have this IL-17 in the meninges produced by meningia IL-17 gamma delta T cells that through a disruption of the barrier, arachnoid barrier, interact with the macrophage to lead to impairment of functional hyperemia. And these two factors together lead to the cognitive impairment that we observe in these mice. So it's a coincidence of two different pathogenic effects. Now, another major risk factor for cognitive impairment is ApoE4, okay? If you are ApoE4 homozygous, you have 12 times the risk of developing Alzheimer's disease. And also, you have a much increased risk of developing white matter disease. So both cognitive impairment caused by vascular factors and those caused by Alzheimer are enhanced by ApoE4, okay? Years ago, we found in an attempt to figure out what could ApoE4 do to affect both of these aspects, all right? We found that mice who express human ApoE4 have an problem with blood flow in the brain, pretty much like the Alzheimer mice did, pretty much like the hypertensive mice did, all right? So having ApoE4 put the blood vessels at a loss. And the mechanism was really mediated by free radicals. So you could reverse the effect by using free radical scavengers, okay? And then using we wondered, therefore, whether these free radicals were coming also from the perivascular macrophage, as we've shown before. But the question is, do perivascular macrophage have ApoE, all right? So single-cell RNA-seq data shows that indeed they have huge amounts of ApoE, more than the astrocytes, which are thought to be the cells, prototypical cell expressing ApoE in the brain, all right? You can see it here. BAM stands for brain-associated macrophages. So the macrophages do have ApoE, okay? So it could be that the macrophage make ApoE, release it by the blood vessel, and then cause the neurovascular dysfunction. So using that mouse I told you before, we deleted ApoE4 from the macrophages by crossing it to a floxed ApoE4 mouse. And what we found that by doing so, we could completely rescue the neurovascular dysfunction of ApoE4. So ApoE4 from the macrophages is responsible for the alteration of the blood flow that we see in mice expressing the ApoE4 gene. Now, it could be that this ApoE that the macrophage make, in turn, acts on the macrophage themselves, who have ApoE receptors, all right, in a cell-autonomous way, to kind of, gear up the system and cause a loop, a positive loop. So to prove that was the case, we asked the question whether the macrophage would be sensitive to ApoE4 when they were exposed to ApoE4. So we patched perivascular macrophages that were identified by injecting dextran into the CSF, and you can see them over here. And when we patched them, we found that indeed, if we put a recombinant ApoE4 on them, they had an inward calcium current, and they also had a radical production, which was blocked by removing calcium from the medium, all right? So the macrophage themselves can be sensitive to the ApoE4, while making also at the same time ApoE4. So the data suggests that there is a cell-autonomous, kind of a network whereby there is an increase in free radical production by these macrophages. So the data in ApoE4 suggests that the macrophage produces ApoE4, which acts on the macrophage themselves to get the radicals going, leading to oxidative stress inflammation and eventually reducing the clearance of amyloid beta and resulting in ApoE accumulation. So now the last part of the talk, which I'm almost done, how is this relevant, all right? I've showed you a lot of mouse, complicated, mouse stuff. What does it mean? So one of the major advances in Alzheimer, world has been the discovery, the development of antibodies directed against A beta. Okay, a number of them have been, and these antibodies were found to reduce the rate at which Alzheimer patient will get worse over time. Okay, and you can see that this over here. When this is one, donanemab is an antibody from Lilly, you can see that there is a reduction in the A beta in the brain. Red means high A beta, more yellowish, whatever, less A beta. So there is a reduction. And with this reduction, there is a slowing down of the worsening of the patient cognition over, weeks and months, all right? So it's not reversing the pathology, all right? It's slowing down by 30 to 40% the worsening that this patient experienced. And this is thought to be the first, a treatment for disease-modifying treatment for Alzheimer's disease, okay? How do these antibodies work? Well, these antibodies work by activating the microglia that chews up the amyloid from the parenchyma, okay? And studies like this one have shown, so these are two patients, before the treatment, this is a control patient. This is a control patient, which has got amyloid, around the vessels and in the parenchyma. And this is a patient who was like this before, assumed based upon the clinical and the imaging and all of that, but then got the antibodies and went from this to that, all right? Less amyloid in the brain, as I showed you in that PET scan before, okay? The microglia is what causes this reduction, but what they don't tell you is that while the parenchymal amyloid was gone, the CAA, the vascular accumulated amyloid, was increasing. So what the antibody do, essentially, they move the A beta away from the parenchyma, but get stuck on the blood vessels. And you can see it here. That while there is this depletion of the parenchymal amyloid, there is an increase in the A beta associated with the blood vessels. And this is thought to be the basis for a major complication of A beta immunotherapy called the ARIA syndrome. ARIA stands for amyloid-related imaging abnormality, and it comes in two flavors: area E, which is edema, shown over here by the swelling, by these MRIs, effacement sulci, swelling, edema, or area H, where you can see actually hemorrhages, microhemorrhages, and accumulation of subarachnoid blood that's called superficial siderosis, all right? So this ARIA syndrome is associated with a variety of symptoms, visual disturbances, headache most commonly, that it can be quickly improved by stopping the treatment. But some patients have died from it. And the patients who have died were patients, in this case particularly, was published. There have been few cases published. In this case, this person had received the last dose of these antibodies and then had a stroke, was thought to have a stroke, was admitted to the hospital, standard of care, was given TPA, tissue plasminogen activator, clot buster drug, all right? And her brain exploded with hemorrhages. So you can see here, those 400 miles of vessels, really gave up, all right? And the patient died. So ARIA can be a major problem, and we try to figure out why. So what are the risk factors for ARIA? Are the three things that I told you about: amyloid beta, as accumulating on the blood vessels, ApoE4, and hypertension. So those three factors that I told you before that have such a profound effect on vascular function through inflammation and oxidative stress are also the major risk factors for this complication of A-beta immunotherapy. So what we may think is that when the macrophages are already primed by hypertension, ApoE4, or presence of A-beta, and then you give them this antibody that releases the A-beta from the plaques, there is a flooding of the perivascular space with A-beta that enhances the oxidative and inflammatory environment, leading to more blood-brain barrier dysfunction and vascular damage that is responsible for this ARIA syndrome. It could also be that the antibody binds directly to amyloid within the vessel wall, creating an immune reaction that may call in immune cells, activating the complement and so on, leading to further inflammation and so on. And so as demonstration, this may be the case, and this is a case who died after ARIA, and you can see the macrophages, around the blood vessels loaded with amyloid beta, exactly like what we would have predicted based upon the experimental data that I presented to you. So then I showed you how the vascular function is really critical for brain health, both from the neurodegenerative standpoint and also from the vascular risk factor standpoint, like hypertension and ApoE4. So these factors, through a neuroimmune mechanism, converge on the neurovascular unit to induce neurovascular dysfunction, which leads to cognitive impairment, perhaps through a neurodegenerative factors by increasing the accumulation of these Alzheimer-like proteins by reducing their clearance, all right, as I showed you, but just by reducing the amount of blood flow going to the brain, which is not good for the brain anyway, all right, or altering the blood-brain barrier. The importance of this is that if you want to kind of treat or prevent Alzheimer's disease, you're going to have to look at both sides of the equation. You cannot only look at the amyloid aspect or the tau aspect, because in those particular patients, there will be also vascular effects going on. So effective prevention and treatment will have to be a coincidence of different approaches targeting both the blood vessels and the brain at the same time. And the fact that if you target only amyloid beta, you have ARIA syndrome and patient may be dying from it, highlight the fact that the vessels have a very important role also in the treatment that you may want to use in patients with Alzheimer's disease. So it's very critical to look at the patient in a holistic fashion and not just on the brain side, amyloid side, or the vascular side, but the two things have been looked at together, both for prevention and for therapeutic purposes. I'd like to thank my collaborators who did all the work across, not far from here, and also the funding agencies for supporting our work. Thank you very much.
Thank you so much. That was a fantastic talk. You're a very good communicator. I think everyone probably enjoyed that in the room today. I did. That was very exciting. So we're going to open up to question and answer period, and we'd like to invite students. If you are interested in asking questions, please don't be afraid. No question is unwelcome. Everything is welcome. And we also have faculty, please ask questions. And our Q&A on Zoom is open for those who are attending remotely. So would anyone like to open the floor with questions? Any students want to go first? No? All right. All right. Dr. Etlinger.
Usually, it's unusual to have 15 minutes for questions. So usually if I'm always a little bit late and we cannot ask any question, which is the best way because you're not put on the spot. Anyway.
Yes, thank you for the beautiful presentation.
Thank you.
It really kind of opens us up to new paradigms and how to really look at all these issues. Very fascinating. So I was just curious regarding the beta amyloid and maybe other factors. Beta amyloid is also synthesized in other parts of the body, other cells.
Yep.
skeletal muscle, platelets, and other places. So I'm just curious if some of the pathologies that you've described on the vascular system in the brain may also be affecting other parts of the body outside of the brain. And reciprocally, maybe there's some beta amyloid that moves into the brain from the other tissues, especially if there's some transient openings of the blood-brain barrier and whatever. And what do you think about that?
Yeah, very interesting. I'm going to give you the short answer, and then if you want, we can elaborate. So the short answer is that the amyloid in the organ is different from the amyloid in the brain. Okay, so if you make this has been done for years. If you make amyloid in the periphery, which you can do very easily, the amyloid doesn't get into the brain. It was a major problem in the developing of the amyloid mice. All right, because they would increase in the rabbits that did these studies, they got amyloid going in the periphery very easily, but this amyloid did not get into the brain. Okay, so that's the story. And also spontaneous amyloid, is a different kind of amyloid. It's got different associated proteins and so on. However, there is a transport of amyloid from the blood, from brain amyloid, all right, from the blood to the brain. Okay, so in principle, so what happened is that as you accumulate amyloid, you release amyloid into the circulation. Okay, now through the lymphatics, which then converge into the venous system, and through the transvascular transport that I showed you through the endothelium. So this LRP1 is the receptor. The RAGE receptor on the endothelium carries it back in. Okay, so in principle, you could have the possibility that as the beta gets out, can come back in. And there is some evidence from experimental evidence that is the case. Except that when you drive peripheral amyloid, you never get amyloid in the brain. Yeah. But so I don't know, so maybe, I don't know if it's like an animal thing or whatever, but it's in principle possible, but what it does exactly, we don't know. Yeah.
Dr. Wolin, did you have a question?
Your insight into the various mechanisms was tremendous and probably very interesting to many people here in terms of the events in the vascular neurovascular interaction and promoting cognitive dysfunction. Something that kept coming to mind with many of these mechanisms as a way of therapy is exercise. And so I thought I'd raise the question, has there been much interest on the components of exercise and how they would affect the cerebral circulation in the various systems, or direction of the systems you've discussed today?
Yeah, so exercise in animals works great. So if you take an amyloid mouse, and you put them on the wheel, it's going to have less amyloid, it's going to do better, it's going to have more neurogenesis in the brain, and so on. Exercise does a profound, you guys know better than I, but there is exercise skins, whatever, there is all kinds of stuff that the muscle makes that has a profound effect on what the brain is doing. So it's not surprising that and certainly the part of the preventive strategy that has been used in Alzheimer's disease. So there is a study called the Finger study. It was a Finnish done in Finland by a woman who now works at the Karolinska. But they did this very rigorous, preventive approach, in which exercise was part of it. In addition to social engagement, stimulation of puzzles and stuff like that, the whole thing, and reduce the risk of getting dementia. Now, what the component of the exercise there was, diet also is another important piece. What the exercise component of that was, no one really knows. But what we know is that if you do these things in isolation, they don't work. You got to do them all together. So presumably in a human, just running on a treadmill all day is not going to make you much better unless you do also the other things that go with it. Yeah.
Yeah, as I just saw many of the systems you were describing strongly influenced by studies and
Absolutely.
exercise, separate though from the cerebral part.
Absolutely, yeah.
So that-
Both cerebral circulation, as you point out, but also these chemokines, stuff that's released from the muscle. Yeah.
Thank you.
Before we go to Dr. Messina, Dr. Guo was given the mic in the back. So I'm gonna let, you want Dr. Messina to go first? All right, you have the floor, Dr. Messina, but he needs a mic. Thank you.
I don't know that I need a mic. I missed how you determined that the reduction in endothelium-dependent vasodilation. What was the test for that?
So with an acetylcholine.
I'm sorry?
Acetylcholine.
Oh, okay, all right. So the standard methodology.
Yeah, in the brain. Yeah, you got to make a hole in the head and
Yeah.
put the stuff on it. Yeah.
Was there any difference in resting blood flow in the DOCA salt animals compared to control?
No.
Really?
Yeah. There was no, in fact, two things which we did not find, which we thought we would find. One is there was no blood-brain barrier alteration, despite the fact that there was major increase in blood pressure, and there was no reduction in resting flow. Now, in the ApoE4, there is a reduction in resting flow. It has been shown in humans too, not only in the mouse, all right? And actually, college students who are ApoE4 positive have reduced blood flow. So, and then when you get older, obviously the whole thing gets worse. Yeah.
What do you think contributes to the metabolic, the failure of metabolic vasodilation?
Well, you mean neurovascular? We call it neurovascular coupling, all right? I mean, the neural activity induced. One component is nitric oxide, neuronal nitric oxide.
So it's a flow-dependent basis?
Is from, NO deficit basis. So interneurons are the major driver, are the neuronal population most associated with the blood vessels that is thought to mediate the vasodilation, okay? In addition to stuff coming from everywhere else. If you think about the brain, how dense it is, But neurons, interneurons do have synapses with arterioles, all right? And one of the mediators of this exchange, there is a COX-2 involvement. Dr. Kaley, I think, worked on prostaglandins, COX-2. There is a nitric oxide involvement. There is a role of potassium ions. So that's why nobody can abolish neurovascular coupling, with a single, intervention. You get back, let's say if you add to a COX-2 inhibitor to a adenosine inhibitor to a nitric oxide inhibitor, you get to 20%. The last 20% is very hard to get rid of. Probably because they are the ions released by doing neural activity that mediate some of that response. So in the case of hypertension, probably it's free radical scavenging of nitric oxide that leads to this attenuation. Yeah.
My last question. What would you predict would be your findings in the spontaneously hypertensive rat?
What would be what?
Your findings in the spontaneously hypertensive rat.
Yeah.
What would you predict?
Well, I would predict pretty much the same thing. And actually, we have studied the mouse correlate of that, which is the BPH mouse, which is again a genetically determined, but we don't know what gene. All right. So we don't use them very extensively because you cannot cross them with anything. All right. You can do genetic studies or, knockdown and so on, because you don't know what the genetic makeup is. Yeah. Thank you.
All right, thank you. Now we're going to go to Dr. Guo.
Hello. Thank you. That was a fantastic presentation. It's an eye-opening for me who's being a scientist, but not in the field, right? So I know you're trying to really open up the field with individual components for your presentation, right? You started with the protein, went with the IL-17, than one with ApoE, right? At the end, one of the slide you presented trying to demonstrate this is really some sort of crosstalk, not a sequential event. Am I correct to understand that? So it's not really from IL-17, I mean, ApoE to IL-17 that leads to the accumulation of the protein.
Yeah. So I showed you components that could potentially play a role in a disease, context. So models, they drive the pathology in a certain direction. They don't give you what the human situation is, all right? So I suppose that there is people who have hypertension, they have increased IL-17, So it's not like totally out of the, of the conceivable. But is IL-17 the only cause of that? Absolutely not. And that's true for Alzheimer's disease, as I showed you. The study in living individuals, over time, they show coexisting pathologies that we don't even understand. How does the coral plexus, leads to now we know that coral plexus brings the lymphocytes into the brain, all right? But how do they, so it opens up new ideas about. And so to go back to the question, everybody's looking for a silver bullet, all right? And everybody says, oh, amyloid immunotherapy, works very little and so on. Of course it does, because it's one piece of the puzzle, all right? If you have 5 or 6 things going on, you have the vessels don't work because the person used to be hypertensive 30 years before, all right? They had a bad diet, was obese because he was a smoker, all right? All of that stuff is cumulatively affecting. The brain takes notes of everything. And at the very end, you have this cadre of things in which amyloid happens to be around, all right? And you have people, 30% of us in this room, I mean, maybe the older kind, will have amyloid in our brain, And why doesn't that cause cognitive impairment, all right? So those are the questions, because so we are talking about a number of factors, working together. And so it's not surprising that amyloid does not make you, go back to when you were 25 years old,
So may I follow up? Since the protein is such an important component, right? Amyloid beta, kind of fitting into my previous research expertise, also plays a role in inducing abnormal angiogenesis, right? In the microvasculature of the brain or any other tissues. And if it does so, I think it's going to somehow in turn leads to maybe increase in perfusion of those microvasculatures, right? What is your understanding of maybe abnormal perfusion can lead to pathology in this case?
So what I was saying, the reduction in neurovascular coupling in endothelium-dependent vasodilation, the reason how the dose may have a negative effect is to reduce perfusion. But the question that nobody wants to answer, what does a 20% reduction in blood flow, as you see in patients with Alzheimer's disease, do to the energetic metabolism of the brain? Do the neurons care about a 20% reduction in cerebral perfusion? All right. If you do, in the old days, they used to put carotid tourniquets to operate people before they had AVMs, arteriovenous malformation. So they used to put, things around the carotids, clamps, all right, that you could occlude. And one, Randy Marshall at Columbia did a study of the cognitive effect of closing the carotids, all right? So and you're measuring blood flow, until you get to 50% blood flow, nothing happens. So what does a 20% reduction in flow in an Alzheimer's patient mean? I don't know. So that's another aspect that we need to really, is that maybe is the clearance effects beyond, it's not really the energetic deficit, but more is the clearance effect, it's the neuroimmune effect on the vasculature. So we have to expand our thinking that the vessels do not just feed the brain, but they are necessary to keeping the whole thing working, Yeah.
All right, thank you. We're going to take the last two from the audience and one from online, and then we're going to cut it off if that's okay. So Dr. Kohler, go ahead.
Yes, thank you very much. Let me ask you, do you see any specific changes regarding the beta amyloid in the hippocampus?
Yeah, that's interesting that you ask, because a student of mine that now is an MD PhD that works in Alzheimer. We did a project, patients who develop Alzheimer's disease a year before they are diagnosed with the disease lose weight, and they eat like horses, all right? So, and we looked at that particularly, and what we found was that there is a leptin deficiency in patients with Alzheimer's disease. And we found that the signaling amyloid affected the leptin signaling in the hypothalamus, leading to a state of kind of, you never felt satiety, So you kept eating, but at the same time, there was an energetic expense increases so that, what you would eat, you would expend and so on. Now, that could be the fact that the hypothalamic neurons may be very sensitive to the effect of amyloid, that could affect this kind of feeding circuit, leading to this, leptin deficiency from the loss of fat in the periphery. Yeah. So very, very important point. Yeah.
All right, last one, Dr. Powers, go ahead.
Hi, thank you very much. I really appreciated your talk and so much research also. Really, congratulations on all the productivity you had. One of the current hypotheses for the 3 monoclonal antibodies that are now clinically approved for use in Alzheimer's disease is the peripheral sink hypothesis. Would you care to comment on that? And that hypothesis is that by binding to and engaging with the amyloid beta in the vasculature, they actually open up the vasculature and also open up the channels for the amyloid beta to leave the brain and thus reduce the toxicity.
Yeah. So what's your question?
That hypothesis, the peripheral sink hypothesis.
Yeah. So that's an interesting point, because if you measure the amount of antibodies that's in the CSF in a patient after immunotherapy, is nothing. It's very, very little. So if you do an experiment in the mouse, you give them the antibody and you measure how much it gets into the brain, it's less than 0.1%. So people are wondering, how do these antibodies work? All right. So one hypothesis is that they get into the coral plexus and then the microglia does the rest. All right. Because if the antibody does not have the Fc segment that engages the microglia, they are not effective. Okay. So there is got to be some parenchymal aspect to that. But it could also be that you have a high concentration of antibodies. We know that A beta gets from the brain to the blood, all right? So you may be, through a sink effect, you may be kind of drawing the A beta out, yeah. However, I bet that within the next year, all of this stuff is going to be mute, because they've developed a brain shuttle, which is a protein that, using the transferrin receptor ligands, is picked up by the endothelium and brought into the brain. And they did that in animals. When you do that, the brain gets loaded with A beta antibodies in a way that you never would be able to get it by simply circulating the antibodies. And so there are 3 or 4 companies who are working on that. There was a science paper published on this that you may want to take a look at, in which they showed it's called, brain shuttle, they call it. And there is a trial ongoing that shows that there is a improvement of amyloid depletion much, much stronger than what you get with the systemic antibodies, and almost no ARIA. okay, which suggests that, the interaction of amyloid with vessel may be very important to the ARIA syndrome, So I bet that all this stuff is going to go away when we have these new devices that bring the stuff in and will, but in the meantime, we got to talk about it, all right? We have.
All right, thank you so much. I think we will close this segment.
There was an online question.
There's a lot of online questions.
Okay.
Would you like to answer one of them?
Yeah.
Okay.
Please.
Then I have one picked out. Irv Zucker has put a question in, and I thought that we would give him the floor since he is well known here. The increase in ROS begs the question why antioxidant enzymes are not sufficient to reduce or normalize ROS. Have you ever measured AOE? Maybe he means ApoE. Or tried to regulate them in either the cytosol or the mitochondria?
Yeah. So in this model, the radicals, may mainly come from ADPH oxidase, from NOX2. The antioxidant kind of field which I'm not extremely familiar with, maybe Mike knows much more than that, is moving from scavenging to blocking the sources, all right? And I think that's the way to go. Because you're gonna, you want to block where there was a cartoon on circulation a few years ago showing, the hydrant, spewing out water, and then there was a little kid trying to get it with a mop, all right? That's the scavenging approach. Whereas if you close the hydrant, so that would be the way to go. That would be what I say. That's why they're not effective, because they don't get into the brain. They're very, very ineffective in terms of biochemically scavenging. And so the future, I think, is in blocking the source of the radical.
Thank you so much. Dr. Wolin, you can take over.
I have the honor of awarding you. I have the honor of awarding you the plaque for today's lecture as head of the selection committee for being the 12th annual Dr. Gabor and Harriette Kaley Endowed Lectureship. Your topic of your lecture was phenomenally wonderful, and it's really been a pleasure having you visit with us. And thank you again for the great lecture.
Thank you very much.
If you want a box, I can give you the box.
That's okay. I can put it in my backpack.
Just bring it ready.
Yeah.
Okay, good.
So my name is spelled even right.
Is that wrong? All right.
No, it's right.
Oh, okay.
No, sometimes, they send it to me. No, when they correct it. They send me the wrong one, and then. Yeah. But when you have someone like him around, I'm sure it's good. Ayatollah, yeah.
But not a real copy of it. And that was where the error was made by the person who makes these beautiful plaques.
Okay, next we have closing remarks by Dr. Edlinger.
Okay, I'll keep this very short, given the time we're at right now. And I just want to thank everybody. And again, this was a really beautiful talk that brought together quite a range of individuals in attendance. We see some not only basic science, but it attracted some of the clinical faculty, which very challenging to get everybody together. And this was a great talk that stimulated a lot of interest across many disciplines. So that's really good. I'll just mention that for the last 3 months, we've been trying to execute the merger of the 3 former basic science departments, including Gabe Kaley's physiology department. And it's been kind of a challenge. I was given the job of kind of orchestrating the merger and trying to make everybody happy. And part of the challenge was to kind of recognize the important history and traditions of the original departments, which is very important, especially a lot of a senior faculty. And this event is really a beautiful way to remember the physiology department, and particularly Gabe Kaley's indelible stamp on the traditions and the great teaching and research that went on in that department. And again, this presentation today really crossed over all the disciplines and the boundaries. So I think a very important event in that sense. And just a personal note, I came here 38 years ago as a very young chair of what was the anatomy department at the time. First thing I did is I changed the name to cell biology and anatomy. And at that point, Gabe was really a mentor to me. I don't know if everybody even realizes this. So I didn't know what I was doing. And he met with me a number of times and gave me very good tips on how to deal with the senior faculty that often may or may not be as cooperative as they could be, and how to deal with administrators. So I never forgot that. It was really very important to me over the years. And I even remember Gabe taking me out to lunch a few times over the years, always in his very fancy Jaguars. That's something I never forgot. And we would have some very interesting talks at our lunch meetings. And as we rode to the restaurants that he would pick to take me to, he always put on the music that he knew I would like, since he knew I was a jazz musician. So he'd have the Miles Davis recordings going in the car. I never forgot that. It's interesting how you remember certain things. So I just want to say to Harriette, Gabe's memory is his blessing. And I think he'll always impact us, and will, because of you, continue this very important event every year, hopefully. So with that, thank you.